So is it possible that do you want me to do the introduction now since Professor Redana is here?
No. You have to do it. You are
Okay.
you are from US. I am from India.
In which case I may have to do, if Deepika allows, I will be the first one to do it, and then
Yeah. I think.
Yeah, I think
So-
we can go ahead. We have 1 more minute.
Yes,
Yeah.
sure.
You start on time.
Yeah.
This
Okay.
anyway.
Suresh,
this-
don't
this
have too many
program-
drinks in the lounge, okay?
Mm.
Although I don't drink at all. I'm just having coffee.
That's what I meant. Don't drink too much coffee.
We don't know what's inside the coffee cup, Suresh.
Oh my.
No, okay. You're excused.
Thank you.
Are you guys able to see my screen?
Yes.
Very
Yes.
well.
Yes.
Very
Okay,
well.
great.
You can start.
Yeah.
I
Divya.
think Loganathan is trying to join, but something is not correct to Logan.
Hope you will join also.
Okay, so do you
Sure,
think I can start?
Deepika, can you please send the link to Loganathan? I think
It's already in the group. I've shared it in the
Yeah.
group.
I think Deepika start.
Yeah, once.
People will join.
Welcome, everybody. Good morning, good evening, good afternoon in your respective time zones. And thank you. And on behalf of the FABA family, I welcome you all to our first webinar of the year on 3D models, a blueprint in drug discovery by Dr. Madhu Lal Nag. And this is our agenda for today. And then going over briefly, Dr. Srinivas, I would like to take this opportunity to introduce Dr. Srinivas Reddy Papayakari, who is the pivotal person responsible for the inception of the FABA US chapter. And Dr. Reddy worked for several years at the Johns Hopkins University as an independent scientific investigator, and he serves as a director of his company, Palamore Biosciences. He comes with a lot of experience in oxidative stress-related mechanistic-based studies related to acute and chronic diseases of the lung. He leads, manages, and develops high-performance teams and fosters a culture of collaboration, innovation, discovery, and cutting-edge research focused on excellence and achieving open communication and continued improved processes in his industry or company. So welcome, Dr. Srinivas, and over to you. You may proceed with the welcome note.
Sure. Thank you, Deepika, for excellent introduction. Good morning, good afternoon, and good evening, everyone. So this is Srinivas Papayakari, founding member of US FABA. So I'm really excited to welcome you all for this wonderful webinar. The speaker is Madhu Lal Nag. So she's going to deliver a beautiful talk with a models, 3D models that will be useful as a blueprint as well as accelerating the drug discovery. So definitely these 3D models are very reliable, scalable as well as reproducible. So definitely these 3D models, whatever they are, have that technology available, that helps. And also this 3D models is alternative to animal models. So I would like to, everyone excited to listen to this talk, and she would walk us through how the 3D model approach is a very good alternative to the animals, and also, it helps in the faster drug discovery. I think with this, not much adding. So I will, go back to Deepika to introduce Dr. Uday Saxena.
Yes, sir.
Deepika, you don't need to introduce me. I will introduce myself and just
Sure,
Sure,
sounds,
sure.
Or
sure, sir,
Faba,
go
yeah.
ahead,
Thank
go
you.
ahead, sir.
Yeah, thank you. Yeah, thank you, everybody, and thank you. I see about 30 odd people have joined, which is good. I'm sure more will join. So my name is Uday Saxena. I'm the one of the executive council members at FABA. I've been with FABA for about, I think about 4 or 5 years now. And by profession, I'm a pharmaceutical scientist. I've spent about 30 years now, both in the US and India, doing drug discovery and, commercializing products. So I just wanted to say a few words about FABA itself and the FABA US chapter. I'm very happy that Professor Redana is here because if you look up if you Google FABA, you will see his picture there. I mean, that's how much he's associated with FABA. Without his efforts, FABA, the way it has flourished, it existed for a long time, but I think it's really flourished with Professor Redana leadership, and very glad he's here. FABA is, as you know, some of you may have gone through the website. It's the Federation for Asian Biotech Associations. Essentially, this was created to cater to the Southeast Asia and foster interaction between different countries and biotech scientists in the different countries. If you go and look up the webpage, you will see that FABA is now in several countries, not just in India. FABA US is the latest entrant to the FABA family. So every country has its own chapter, but the corporate, the headquarter chapter is in India. And let me tell you a little bit about what FABA does. FABA is essentially a think tank as well as a facilitator for all kinds of scientific interactions. Firstly, I can tell you that FABA, so why should you be interested in FABA? Let me just focus on that. Number one, if you go to the parent FABA website, you will see something called Executive Council. There's about 25 to 25 or so very experienced members in the Executive Council, both from academia and industry, who will serve as a wonderful resource for you, whether it's academia, like I said, or industry, or biologics, or drug discovery, you name it. There is a leader among the Executive Council. There is a leader who you can tap into and use their experience, their contacts, and their mentorship to help you drive your own career. So I would say that the number one thing that FABA brings to the table for the global US global chapters is the outstanding executive council that has been put together by Professor Redana. I have not seen a more diverse and more experienced.
Sir, can you unmute
Sir,
your?
you went on mute, sir. Saxena, sir, you went on mute.
Okay,
Unmute,
when was
please.
that?
Yeah.
Did I go on mute long time back or just now?
No, just maybe 10
Okay,
seconds.
yeah, I was talking about the Executive Council. I think these are, as I said, opinion leaders in different fields who are willing to give their time to help you in any which way you want. So I would say that's the number one reason why you should really be actively interested in FABA and tap into these people. I mean, there are people here who have spent both time in the US and India, so very familiar with how academia and business works on both sides of the world. The other thing is the knowledge that it provides. I mean, case in point is today's seminar. FABA actually champions many such wonderful seminars, workshops, competitions for awards. So I think that's the other thing you should look forward to is a lot of learning that comes out of the things that FABA conducts. And this is whether you're an experienced scientist or a startup scientist, you will all benefit from this. And just to tell you, we have a flagship program called the Drug Discovery Program that usually held in 2 weeks in August. I think the advertisements have gone out. If you're ever interested in, in the industry, this is a must attend function for you, because end to end, starting from what is drug discovery to how to do a clinical trial to how to commercialize a product, you will hear from opinion leaders over those 2 weeks. So the second reason, as I said, is all of the educational help that you'll get. The third and last but not the least is networking. I mean, whether it's in the US or India, if you ever wanted to either collaborate with somebody or do a startup in US or do a startup in India, be an entrepreneur, essentially, there is also a wing dedicated to FABA entrepreneurship. So besides the learning and development I talked about, a big push at FABA is to help emerging entrepreneurs. Like I said, if you wanted to set up a startup in India, I think FABA can help you tremendously in getting you connected with the right people. So these are just the three things I'm talking about. And I have to tell you that the startup ecosystem in India is exploding. So just even whether you may or may not want to start a startup in India, but just learn from it as to what's going on in that country, because I honestly believe that what the US saw in the 80s and 90s, that startup culture, which I don't notice that as much in the US anymore, but India is just exploding. I mean, there's so much activity and the buzz that's going around. Just being connected with that will give you a lot of ideas, contacts, and also, I don't want to take too much of your time, but very happy to be able to say a few words. Please contact me if you need. I spend my time between US and India. So just let me know if I can be of any help. Again, taking the opportunity to thank today's speaker today and all of you for joining. One last thing is, please pay the membership fees, because we are a not-for-profit organization. Pretty much all of us work as honorary members, but there are some critical members such as Deepika and all who need to be paid for all the hard work they do. And the only source of income is your membership. And if you look at it, it's less than $1 a day. So for less than $1 a day, you're going to gain so much. It'd probably be well worth your time and effort. So that would be my last plug for FABA. And Dr. Madhu Lal Nag, I very much look forward to hearing your presentation, and I'm going to stop here and hand it over to Deepika. Thank you so much. Yeah.
Thank you, sir. And then I would like to take this opportunity to discuss a little bit about Dr. Raghu, who is also presently working for Inspiro. He is a scientist who turned into a seasoned sales and business development executive with last 20 years of experience in the field. He widely networks with professionals in the academia, biotech, and pharma industries, actively mentors and coaches scientists who are transitioning into non-academic, non-bench-based research careers. And he's on board of multiple nonprofit organizations and biotech startups, and also interested in entrepreneurship opportunities. Presently, he's also serving as founding member of the FABA US chapter, and he's doing an amazing job advising us and pitching his time and effort into this organization. Welcome, sir, and over to you, and you may go ahead and introduce the speaker and Inspiro. Thank you.
Thank you, Deepika, for the very good introduction. Nice introduction. Thank you. I'd like to take this opportunity to introduce Dr. Madhu Nag, currently the CSO of Inspiro. Although we've been colleagues for a little over a year and a half, I've known Madhu for a really long time. She used to be one of my customers from one of my previous jobs as well. So awesome person, very glad to introduce her. I'd like to say a few words about her background. Madhu is a scientific and executive leader with a profound expertise in developing preclinical MPS and 3D models targeted therapies, biomarker discovery, and the application of cutting-edge technologies across various domains, including toxicology, safety, pharmacology, oncology, functional genomics, and stem cell research. With extensive training in both scientific and business realms, Dr. Nag possesses a unique blend of skills that bridge the gap between academic research and translational science. Dr. Madhu holds a PhD in molecular and cellular oncology from George Washington University and a master's degree in bioscience business from Keck Graduate Institute. Her primary goal is to unify the academic and translational facets of pioneering science in oncology, metabolic diseases, and investigative toxicology. Leveraging insights from contemporary therapeutic regimens, Madhu creatively translates state-of-the-art research to promptly address patient requirements. I'd like to comment on a few strategic achievements and corporate growth accomplishments of Dr. Nag. She's been marked by remarkable achievements, including building excellent preclinical translational research capabilities from scratch to guide patient indication, selection, and combination strategy in both adult and pediatric oncology, leading efforts to develop and implement strategies for clinical biomarker testing, patient stratification, combination therapy, mechanism of action studies, and drug candidate differentiation. She's developed and maintained and grown external research partnership to leverage external resources to grow nationally and internationally. She's established platforms for regulatory research to be communicated externally, thereby emphasizing greater transparency in research communication to benefit public health. She's written and awarded multiple grants by NIH and other institutions. She's initiated the formation and managed activities of the first 3D standards working group across multiple groups representing industry, academia, and government to develop consensus in directing research programs and focus on building representative and prognostic 3D models for oncology programs. In addition to her role at Inspiro, Dr. Madhu Nag holds concurrent appointments and has received numerous accolades, including being a co-chair and scientific advisory board on Fight Cancer Global. She functions as a liaison between NIH, FDA, and the broader biotech and pharma community. She's bagged multiple awards with the FDA, CDER, NIH DDIR, NIH Director's Challenge Award, Intramural Training Award, et cetera, the list goes on. Overall, Dr. Madhu brings a wealth of experience, expertise, and a passion for advancing science and disease management with a strong desire to contribute to transformative advancements. She seeks to establish long-term associations with visionary organizations poised to make a meaningful impact on society. I couldn't think of a better person to invite to present the inaugural address, inaugural webinar at our FABA US chapter. Welcome, Madhu, and I'd be curious and I'd love to listen to your presentation.
Thank you very much, Raghu. Thank you very much for the very generous sort of introduction. And now I'm a little stressed out about having to live up to it. So Deepika, may I go ahead and share my screen? Right here, sorry. Here we go. Let's see if I can share this. Can you see my screen, everyone?
Yes.
Perfect, wonderful. Well, good morning, good afternoon, and evening, again, depending on where you are. It's an absolute privilege to be here today to talk to you about 3D models and sort of how I've seen them evolve over the past 17 years when I first started my foray into this. I will not be giving you what I normally do, which is a sales pitch for our beautiful 3D models in Sphero, but either Raghu or I would be happy to share all of that information with you. Because of the focus on drug discovery and a very sort of dynamic spread of audience today, I thought I would focus today's talk really on the different perspectives that scientists, academicians, industry, regulators all bring to this, where we are now with 3D models, why we should be using them, where we should be using them in therapeutic development. So it's much more, there's a lot more science in today's talk, because I also wanted to give you a real world example of how we developed a patient-derived 3D model to come to a drug that is now, in the midst of an IND submission, and how long it has actually taken to get there for a difficult to treat disease. So I thought I would begin a little bit by giving you an agenda for how my talk is structured. I always now begin with the patient, the destination, right? It's the patient. We've traditionally in preclinical research always focused on cell lines and the tools, thinking about the patient more as the end user. But I would say that over the last 17 years, if there's one thing I've learned, you sort of have to start with the patient first, design the models for the disease that you are going after, really understand the context of biology, and then do the experimentation. Following that, the format of my talk will be a little bit, about the trip advisory, the journey itself, something that I couldn't emphasize enough, which is the role of team science and complementary expertises coming together in therapeutic development, and then of course the role of innovation in therapeutic development. I'd like to begin a little bit with telling you a little bit about myself. I do not have a very traditional career path, so to speak, and people like Raghu can really speak to that. But what I have had has been a tremendous learning experience, and it continues today as well. And what I wanted to highlight was what I've learned over the past many years from each of the places that I've been at. So as Raghu mentioned to you, I got my PhD in 2009 from the George Washington University in DC. And, I did a very traditional molecular oncology PhD looking at the role of PI3 kinase in lung cancer. And my big learning from that as a very, very academically driven student was to break down each question or problem into bite-sized pieces so that you could look at the whole much better. Sometimes, you get a little, or at least I used to get a little overwhelmed by, the enormity of experimentation that had to be done to answer a particular question. But I really left with that one learning that really stood me in good stead for the rest of where I was going to go. I then did a postdoc at the National Institute of Aging and the Sidney Kimmel Cancer Institute at Johns Hopkins, which is where I actually started my work in 2009 on 3D models. At the time, it was in ex vivo models. And if there's one thing that I learned then, the throughput was 48 well, 96 well, was that every model was different. So depending on the kinds of models I was developing, I'd get a slightly different answer. But when it came back to the basic biology of the cancer that I was studying, which was high-grade serous ovarian cancer, there was some very universal truths. After having finished my postdoc, I went on to the National Center for Advancing Translational Sciences, NCATS at the NIH, which is actually where I met Raghu. I was initially a scientist developing high-throughput screening models, and I'll talk a little bit more about that later on, for cancer on the small molecule space. And then I went on to become the director of the Trans-NIH Functional Genomics Center. And that's truly where I had a beautiful understanding of the fact that I was just a very small cog in the wheel of drug discovery. And it took multiple different disciplines actually answer even specific questions. So as a research biologist, I worked heavily with chemists, with engineers, with informatics scientists, and really got my first taste of drug discovery, but in a nonprofit environment. But in about 5 years, I also realized that I had a big gap in my knowledge, and that was that I didn't fully appreciate how important the regulation of therapeutics was. So I went on to the FDA. I was with the Center for Drug Evaluation and Research, where I learned a lot about the safety aspect of it. So my big learning from there was that safety and toxicology and efficacy cannot sort of be studied in silo. And then I went on to Day One Bio before moving to Inspiro, where I, for the first time in my life, actually did real life therapeutic development for a rare pediatric disease. And one of my biggest joys has been about 2 weeks ago that drug was actually approved for use in the US for pediatric low-grade glioma. So it was a tremendous experience. We were a very small team, but the drug is now approved for use in children. So beginning now with the destination, right? The destination being the patient. As I mentioned earlier on, we're traditionally not used to thinking about the patient as the destination, but this is one thing that really came to bear during my time in the translational science realm, which is that not only in basic research, but in all realms of research throughout the drug development process, and we'll talk a little bit about it now, keeping the patient front and center becomes very important in any preclinical model that we develop. And this then gets down to really understanding the drug discovery process. It's, most of this audience needs no introduction to the fact that it's a very long, drawn out process, taking multiple years and a lot of money, right? But I think if you look over here where we would start looking at when do you switch to what models, when do you use 2D models, when do you use 3D models, this is the part of therapeutic development that takes the longest time, preclinical trials, right? And it also is the area where you have the most attrition in the number of molecules or therapeutic entities that you develop. So the idea really here for us is within target identification, within in vitro studies, to keep that patient in mind so that you develop the right preclinical model, and we'll discuss in a little bit what the right preclinical model is, so that you have a much higher chance of getting to the clinical trial, even to phase 1, than you would if you use sort of a very generic 3D model. As I sort of showed in the previous slide, a lot of us think of drug discovery, the continuum of drug discovery as starting here with drug discovery and preclinical studies, but actually it starts now in a state at a very academic stage. And this is what I meant by bringing different perspectives into drug discovery, because now a lot of small spin-offs are coming from basic research, clinical observations, which then go into a medchem process where you're really developing and identifying the therapeutic agents. You're doing concerted target ID and target validation before going into phase 1 and phase 2. And I would argue really that the choice of the right in vitro model starts here in your pre-discovery space, whether you're in academia or in industry. So then it comes down here. And coming back to the continuum, really understanding the model, really understanding the patient, and really understanding the molecule or whatever your modality might be. I will warn you here that this talk is predominantly oncology focused, but the lessons from here that are more translatable in terms of choosing the right model really, and we've seen, with a lot of our customers that Inspiro really span the spectrum for toxicology as well as many, many metabolic diseases. So it's really about the bigger translatable learnings and less about sort of the actual use case of the biology. So in terms of then keeping the patient sort of front and center, one of the first things that I've done in my career was to develop with respect to 3D was to understand how you would build an intuitive screening platform for discovery, right? So it really starts then with understanding the biological question of interest. And we'll talk about this a little more because with 3D models, the higher your chance of success, your chance of success is much higher if you're choosing developing a model that answers a specific part of your question and not sort of a catch-all for your entire disease phenotype. So you start off with the right right biological question, then design the right physiologically relevant models. And this is usually sort of like a Lego building block model where you develop high throughput screens for simpler readouts, and then, less high throughput or medium high throughput screens for more simple phenotypes. These screens, all of the models that we develop are usually or have developed in the past, can be run in high or through medium put, depending on what you want to see. Automation is always an excellent, is usually more a friend than a foe, especially if you have a big medicinal chemistry outfit or you have a lot of modalities that you're testing. I would also want to stress here that because of polypharmacology and associated with many drugs, one of the biggest things that I learned was to be able to look at multiple perturbagens to identify targets and interrogate the function of your target instead of just using one modality alone. For any outfit that's looking at discovery platforms or even toxicology, having an in-house informatics group is extremely important, because if there's anything I've learned, it's also that you need someone who's unbiased to look at the data. There have been many times when I've been wanting my data to say something and it doesn't, and then it's a little bit depressing, but you actually need a biologist to give you the right target and the right gene to go after. So having sort of given you an overview of where in the drug discovery process we would look to switch from 2D to 3D models, I'd like to start a little bit with then sort of continue a little bit with the trip advisory down this lane of developing 3D models. So the big question really is why? Why are we going into 3D? And the answer here is reasonably obvious. Human beings are not two-dimensional. So if you want to model the patient, if you want to model the person you're going to use the drugs in, you have to develop or recapitulate that biology in vitro as well, which is sort of the biggest reason for going into 3D models. The other aspect of looking at 3D models really is when you develop a 3D model, you kind of have to know what biology in a human being it is going to model. So understanding the continuum of going from 2D cultures where you're looking at things that are flat, every cell sits really well and has sort of no diffusion effects with the drug getting to it, going into 3D organoids where you then develop more complex biology and multiple cell type interaction, but without the influence of shear force, stress, and flow. Going into then your traditional microphysiological systems where you now introduce flow and shear stress, which would emulate the blood flow in your body and the ability for a drug to diffuse through multiple gradients to get to its target in the tissue and to its actual sort of like the destination of the patient, where you have multiple physiological inter-organ interactions that also affect the ability of the drug to interact with its target and then to have its biological effect. So I think the advantages of understanding how you go from in vitro to in vivo and establishing that before you establish your preclinical 3D model is extremely important. Now, I will say here, all models are wrong, right? Again, because we go into thinking that something's going to be a catch-all, something's going to give us all the answers. That's not the case at all. All models are wrong, but many of I don't want anyone to go home with today is to think that 2D models are useless. They are not at all, in fact, especially with iPSC-derived models for very specific target engagement studies. 2D models are extremely useful tools, but it has to be used as part of a continuum of other models. I think no model in its sort of in exclusion gives you all the answers. So as you think of the continuum of models, you have 2D models, you have spheroid models, you have organoid models, you have printed tissues, and you have organ on a chip. And the one thing you might want to keep in mind here is that as you increase physiological relevance, your ability to increase throughput decreases. So for you, when you're answering a question, the dependency really is how much throughput, what kind of output do you want, and to organize sort of the complexity of your model keeping that in mind. One of the biggest things that the 3D community still grapples with is how do we actually standardize the data that comes out of spheroids, organoids, and printed tissues, because this is sort of the big lift in the 3D community right now. And I can tell you that a lot of work and thought process really has been going into technical standardization of this. So what the FDA has really been pushing in order for them to qualify these 3D models so that people can adopt them is really to technically standardize all the plates, all the cell types, all the endpoints and the relevant tissue function that come from the 3D models, to develop quantitative assays to be able to quantify disease phenotype that you normally can't quantify. It's just something that you see by eye under a microscope. And then most importantly, for the regulatory authorities, what becomes very important is to be able to pharmacologically benchmark these 3D models against control compounds that are already in the clinic or are already on the market, to see at what point do the 3D models separate out from the 2D models in terms of being able to predict the efficacy. Now, in terms of 3D models, where are they being used in in therapeutic development? So one of the big areas that we find they're being used is in investigative toxicology. And this is extremely important because, as I mentioned to you before, I think one of my biggest learnings really was it doesn't matter how good your drug is in engaging its target, engaging its efficacy, completely causing tumor growth inhibition or tumor suppression. If it's not safe, it doesn't matter because it cannot be put into a human being. So the many groups that are doing investigative talks are that really look at the de-risking of molecules if there's a safety flag, have been looking at using 3D models instead of going into the traditional non-predictive animal assays, as you can see here, so that they're able to be to inform on more of the mechanism of action for why a particular modality is toxic. One of the major goals of investigative toxicology is to be able to improve preclinical decisions and not just keep cutting drugs in the pipeline just because there's a flag without understanding why there is a safety risk associated with that. Another big area where we see the use of 3D models again is in tumors and 3D spheroid models, because traditionally only about 8% of cancers are mutationally driven, right? So there's a whole 92% of the patient population that not only differ demographically or have sort of other comorbidities, but they also do not have a driver mutation. And it's in for these patients really that we have to think about really modeling their tumors in with all the associated pathophysiological gradients, drug concentrations, comorbidities in a plate to be able to deliver sort of really get to precision medicine with these people suffering from cancer. So from both the perspective of people developing the models as well as the regulators' perspective, there's three big pillars, I would say, that you have to keep in mind. One is to define the context of use for any 3D model that you want to use, to make sure that it's standardized against the disease and the standard of care, and then to be very, very particular about what the biological question you're really asking is, because no 3D model is going to answer all the questions. If you want to get the efficacy at the end of it, you need to be able to develop a slew of models. I will pause here and see if anyone has any questions before we move forward. And if not, then I will move to the next section of my talk, which is the journey. And this is where I'm going to a use case which took, I would say, from 2013 2023. So it's been a 10-year journey, starting with the biological question, developing the right 3D model for it, really refining the medicinal chemistry, and then submission of an IND. So this this particular use case is focused on high-grade serous ovarian cancer, and more importantly, it was a drug. The idea was to develop a drug that inhibited metastasis. So this was not to be able to kill a primary tumor, but it was to be able to prevent the metastasis of high-grade serous ovarian cancer, for which the survival still remains abysmal, to its site of metastasis, which is the omentum in most women. As most of you may already know, without the details, obviously ovarian cancer is still a leading cause of death from gynecological malignancies worldwide. And the biggest reason for this really is that most of the early stages of ovarian cancer have no distinguishable symptoms. So like most things, they manifest with GI symptoms. So by the time you actually go in for diagnosis, you're either at stage 3, where it's left one of the ovaries, or at stage 4, where it's already metastasized to the omentum. The risk factors for ovarian cancer, obviously, there's a high genetic family history risk. So a family history of either breast or ovarian cancer, mutation in the BRCA1 or 2 genes, or Lynch syndromes. So for us, when we started sort of going after this in 2013, it was really important to mechanistically understand the tumor and its microenvironment to be able to correctly develop a model for this. What did understanding the microenvironment really mean, right? So over what you have on the right-hand side over here are 2 ovarian cancer cells interacting with each other and the tumor microenvironment, which is the metastatic niche, and that's the omentum, which lies underneath the diaphragm in all women. Traditionally, all of the therapeutic screens that were run using these 2D sort of monolayer assays, and it was typically not something that was giving us anything that was predictive. So the play was to develop 3D models that more accurately represented the interaction between cells that were not sitting on a plate, and then to develop even more predictive models that represented the interaction not only between cells in 3D, but between cells and sort of their substratum or the microenvironment. So what we did then really was to move towards a high throughput translational platform, where we focused on developing 3D models, number one, just for solid tumors itself. So treating the cancer while it was confined to either one or both ovaries, or then developing a model for high throughput metastasis. The reason for this really was, with the traditional models where you had sort of the clonogenic survival assays, there was a high attrition rate of compounds going into animal models, and then an even higher attrition rate of compounds going into the clinic. So the idea really was then to develop models that would give you a high success rate of compounds getting into the clinic. What did that really entail for us in terms of throughput? As I mentioned earlier, as you increase throughput, you decrease physiological relevance. So for us, again, it meant really developing a slew of assays from 2D to 3D spheroid to 3D organoid in increasing physiological relevance, which would answer different biological questions about primary tumor shrinkage as well as metastasis. The first thing that we had to do, which anyone has to do in high throughput screening, was really develop this in a 3A84 well plate, develop the cell lines. So for this model, in fact, we started off with cell lines and then moved to patient-derived cells. So with the cell line models, what we did was we developed a fluorescent assay. This was to mimic the primary tumor assay. And as you can see, these is, as I mentioned earlier on, one of the big things that the FDA wants to see from 3D models is technical standardization. And the first thing that we had to do was develop a robust and reproducible assay to show that from run to run to run, all the different readouts correlated with each other. Once we sort of established the technical aspect of it, we went into the biology. So what you have here on the left-hand side are fluorescently labeled ovarian cancer cells. And then as a counterpart, we also did pancreatic cancer to basically show that, you know, while you do the same experiment, every model behaves differently and has to be tweaked for its own biological question. So as you can see here, one of the big things that we focused on very early on for the solid tumor models was development of multiparametric responses, because the morphology of the tumor can tell you something, but the readout can tell you something else. Understanding that interplay between morphology and the biochemical endpoint is extremely important, because it gives you a much bigger picture of what's actually going on in the solid tumor model. This is better visualized here when I talk about the importance of really understanding multiplexed chemotherapeutic responses. If you look at the panel on the right-hand side of the screen, right, for the two different drugs, selenomycin, vorinostat, both chemotherapeutics, both different mechanisms of action. If you look at the panel on top, you'll see at 50 micromolar, it completely decimates the organoid. But a separate drug, vorinostat, at 50 micromolar, it completely keeps the solid sort of organoid intact. But if you look at the biochemical readout on the left-hand side, they have very, very similar IC50s, right, potency. This becomes important again in going back to understanding the biology of your tumor, because if you're able to see a lack of viability like you see with vorinostat, but you see an intact organoid, this is very reminiscent of primary tumors that are able to regenerate because they have a quiescent cell population within the organoid itself that is not in fact touched by the drug. So being able to understand, again, add in multiple responses to be able to understand the effect of a drug in 3D is much more complicated than it is in 2D. It's also much more important so that you understand the mechanism of action of your drug. Again, this is yet another example in ovarian cancer cell lines, a cell line primary tumor model of looking at different drugs and how different readouts can show you different things. And for each drug, the idea really was to develop a blueprint, which we did for a panel of 38 clinically proven compounds, a blueprint of the different morphologies as well as the biochemical readout, so that we could get a better understanding of how these drugs interacted in 3D. The next step really was to go into 3D models, which were multi-cell type, again, much more representative of an actual tumor. And this is when my tryst with Inspiro actually started 10 years ago, where I, working in Rockville here in Maryland with my partners in Schlier in Switzerland, we developed a multi-cell type tumor platform with fibroblasts and epithelial cells. And as you can see here, both from the immunohistochemistry on the left, as well as the therapeutic responses on the right, we were looking at two different effects for this panel of 38 drugs. We were looking at time. So over time, what do you see with response to therapeutic effect? And then more importantly, when you add in the multiple different tumor cell types, does the response change? And as expected, it does, right? So I think that was a very important learning from many years ago, that having the right cell types in a model is understanding its therapeutic response, because the response is not a function of just one cell type. It's a function of the tumor microenvironment. And as you can see, like I mentioned over here, going from homotypic to heterotypic tissues, you see a huge shift in the potency of these drugs. So having sort of understood from the primary tumor models that the tumor microenvironment was very different, this was a seminal study that we did with patient-derived cells. So I had a collaborator at the University of Chicago, Dr. Ernst Lengel, who was a very forward-thinking surgical gynecological oncologist. And what he did for me was during surgical debulking for high-grade serous ovarian carcinoma, he collected all the ascites, which is the fluid in the peritoneal region, and separated the mesothelial cells and the fibroblasts from 20 different women for this particular high throughput screen that we did. And the object really here, as I mentioned earlier on, was to identify the inhibitors of metastasis to the site of metastasis. So we used cancer cell lines, but then we used fibroblasts and mesothelial cells from these women in a 1536 well dish and really recreated the tumor microenvironment that was relevant to the patient biology for this particular assay. As with anything else, obviously the first order of business was to really technically characterize and standardize the assay, which we did in 384 and 1536 well formats. Then we optimized the biological control. And as you can see over here, tomatine, which is actually a natural product present in tomatoes, was the best biological control and then was used as the assay control. This particular 3D assay, which consisted of this multi-layered set of cells, was run as a high throughput screen for a GFP readout. So basically you create the omentum in a 1536 well plate. In each well of a 1536 well plate, you add the ovarian cancer cells as on top of this, you allow them to adhere, you wash it out after they've been treated. And then the bottom line is, if you see adhesion, then the drug did not, or if you see a GFP readout, the drug did not inhibit it from adhering and metastasizing. This was a beautiful screen because for all of the first test compounds that we tested, not all of them, for the ones that came out as hits in the GFP fluorescent non-cellular assay, we were able to validate and confirm hits from this 3D cell line based screen to a mouse model. So using different ovarian cancer cell lines at the same assay, we were able to reproduce the same results for all of them that came up as hits in that particular assay. In addition, as I mentioned earlier on, it's important to have different validation for different aspects of biology, especially when growth inhibition and tumor regression. And here again, we found that from the 3D screen that we ran, the same compounds that were coming up as hits in that assay were also extremely efficacious in preventing in vivo adhesion or invasion as well as metastasis. And then this in turn correlated with a much better chance of survival in these mice. So really, in terms of if you look at the progression of cancer, going from a 3D in vitro screen with patient-derived cells, we were able to mirror the entire spectrum of what the effect of lack of inhibition in vitro would do to invasion, adhesion, and then ultimately survival. This screen then led to a 100,000 compound screen across multiple different libraries, which we ran the Citravon, the NPACT, beta-acin analogs, and some oncology compounds. And we're really, what was phenomenal about this screen was that even though we used cells from 20 different women, the targets were unmistakable. We weren't getting hits for the particular targets from all over the place. And unfortunately, I can't disclose the target right now, but the beauty of it really was that despite the fact that we used all those patient cells, the biology was unmistakable. There is one clear driver in high-grade serous ovarian carcinoma, and we kept hitting it with sort of different clusters of compounds. We had some fantastic medicinal chemists who then went into further improve the different, one representative from each cluster of the compound. And then these were then further put in through secondary confirmatory screens. And again, most of them performed beautifully as well. And as you can see, we ended up with two compounds here, the PP121 and the milcyclib, that we took in for further characterization in ADME models, in other in vitro models, and other in vivo models as well. Again, PP121 and milcyclib did very well, as did the control compound. So we ran them across multiple panels to make sure that they were not hitting other targets. So we ran many panels with reaction biology to look at off-target kinase activity, etc. We then did a lot of SAR, which is structure-activity relationship, for the two compounds that we had against multiple cell lines, against multiple in vivo models with the cell lines and GEM models as well. And again, the compound that we ended up selecting to go into an IND was reproducible again and again and again with different patient models, different confirmation screens, really then speaking to how robust the biology was. And we ended up submitting an IND for this indication. So what I'd really sort of like to end with is that drug discovery is a very, very long journey with a destination. So the good thing is there is a destination, but if there's anything I do want to emphasize, it's that collaborations are a must for novel drug discovery. Being able to interact with regulators ahead of time, especially when you're using new in vitro models, is extremely useful. You know, they actually don't live in a black box, unlike the impression that most people have. And then the other big thing that I would like to sort of emphasize is that it's absolutely essential to embrace new technologies like 3D models. But as with anything, you trust but verify, right? You know that it brings something innovative to the table, but verifying the in vitro to in vivo translation is extremely important for the use of any models, and that sort of goes to AI models as well. And then, as always, every new innovation begins with an unmet medical need, and this really took a team across the U.S. with different institutions to come together to be able to put together a 3D assay that eventually identified a compound that's going into an IND. So with that, I will stop and thank you very much for your attention, and I'm happy to take any questions.
Thank you, Dr. Madhu. I think that was an amazing presentation and great data that you showed. So I have a small question. Can
Sure.
these 3D models be used to study things like metastasis?
Absolutely. And, you know, Deepika, like you said, I think it comes, so now more and more models are being used to, let me take a step back from there. I think the realization that you have to model metastasis differently from primary tumor models is really coming to a head. So we're really trying to develop very separate models to answer those two biological questions.
Okay. Great.
Madhu, it's very interesting and exciting. Suresh here.
Hi, Suresh.
Yeah, thank you. Great presentation. One question, Madhu. When you did the small molecule SAR and so on from the 3D spheroids, from the wells, apart from staining the spheroids, did you collect the supernatant and estimate cytokines or something mediators like that, biomarkers?
Yes, exactly. So I think you know, for the 3D, I only showed sort of the primary cell, but the primary screen. But then when we did the confirmatory screens, there were separate in vitro adhesion assays. There was a cytokine profile because we also added in the confirmation screen adipose tissue. So we were looking at the inflammatory signature, which is high in ovarian cancer. So you're absolutely right, Suresh. You know, that was also taken into consideration.
Thank you. Thank you.
So the cytokine estimation was you like a multiplex one or just like, you know?
Correct,
single,
exactly. Because with the confirmatory screen, Dr. Reddy, it was the throughput was lower. So it was 96 wells. So we were able to pool supernatants and then look at their profiles.
So was it like, you know, 9 cytokine or what is the profile number of cytokine? Like, how many cytokines in that profile?
We
Any idea?
profiled six cytokines.
Okay.
Yeah.
Thank you.
There was another question.
Sorry, Kalyan. Yeah.
I have a question. Hi,
Kalyan,
Madhu, this
hi.
is Kalyan. Indeed, it's a wonderful presentation. I love it. And I am also from the Johns Hopkins family. I have 2 questions. The first question is that, is there any possibility, I'm also working in the anti-cancer drug discovery. Is there any chance of go forward because of some of our molecules are in IND studies? Is there any chance of, you know, collaborating with your company? That is one question. And the second question is that, is there any models for the CNS central nervous system, particularly for the Parkinson's? Because first I did my PhD in Singapore, one of my collaborating professor who already developed a target for this Parkinson's that is in IND studies actually. If there is any models with you, we already have a preclinical drug in our hand. Next, we are going for the IND followed by the clinical trials. I'm taking Reddy's help too. So these are two questions. I can talk to you privately with Reddy.
Yeah. So
That
any
would be,
model
no,
that can be-
absolutely, yes. Yep. Go ahead.
Any model using your 3D model for the Parkinson disease screening, right? So that's what he's asking.
No, it's a question. So we do have a neural model at Inspero, and then we're also partnering with a company that can actually do a lot of imaging in these spheres. So we sort of do the bigger screen, and then they do a lot more feature extraction from an immunofluorescent model that has that has correlation with clinical disease features. So we can absolutely talk about that.
Sure, sure.
And we are planning to write some SBI grant for the September intake. So we
Mhm.
will discuss if you know, it is good for the pharma too, and we are thinking of go for the ELD. If you want to do some studies for our company, our company actually, that is US company. So if you are interested, then the next inputs, right, Reddy will discuss with you.
Sure, absolutely. It would be a pleasure.
Thank you, Madhu. Thank you.
Dr. Madhu,
Yeah,
wanted
also
to
can
ask-
go.
Dr. Madhu wanted to ask, this is quite fascinating, wanted to ask whether you used an organoid model. The patient cell work that you talked about, you alluded to adherence and then
Correct. It was a
viability.
layered model. Correct. Yes.
So it was not a spheroid or an organoid model.
No, the primary tumors, Jugnu, we did with a spheroid and an organoid model, but the metastasis assay, as I described it, was done with in a layered manner. So we recreated the omentum, which was mesothelial cells in a particular ratio. They secreted the collagen, and then we added, we tried to recreate what metastasis would look like, which would mean you would have migrating cancer cells that would then attach to this. There is a second variation of that experiment we did that I did not present, which was we had spheroids metastasizing like they would as in the ascites, which is in the peritoneal fluid, and had the spheroids attached to the omental layer. But what was fascinating there was, like I said earlier on, the target for high-grade serous ovarian cancer, it made no difference whether the cells were migrating as a monolayer or as a spheroid. What made the difference was the fact that we had the 3D omentum available for them to invade and metastasize through.
So just a follow-up question. I know several people have questions here. So when you went for screening the 100,000 molecules, those libraries, it was at Vertex. So we've had screening for 1 million compounds there.
Yep, right.
So at that point, was an organoid possible or a spheroid? Or were you using simpler cell line based assays to triage through such large number of compounds?
That's a great question. So we did have a counter screen built in, which was a spheroid screen for growth. And just to make sure, because like I said, when I showed that cluster of compounds, one aspect of it was, showing, like, if you remember, so there was an efficacy seen with inhibiting adhesion, but then there was also a correlative sort of like viability effect. And what we wanted to do for the tier 1 compounds was only to find compounds that would inhibit adhesion, but not affect proliferation, because we wanted to use it in combination with an anti-proliferative agent. So we ran 2 screens. One was the adhesion screen that I presented, and then one was a spheroid viability screen.
Okay.
I see some questions in the chat. Deepika, can you? Please. Someone asked, computational.
Okay, so I see here we go. Let me go down from, so I have a question from Kiran Kumar Nalla. What computational resources are needed for developing 3D models? I can actually send you, so we had some phenomenal informatics scientists. In fact, one of them, Raj Guha, he went on to Vertex. He developed, and because at the time it was NIH, it was open source, he developed a whole open source way of looking at now from an imaging endpoint, there are multiple companies actually, PerkinElmer being, I think, the one with the biggest strength in this area, to look at multiple layers or slices of these 3D models with different imaging capabilities, and then developing a 3D representation of that spheroid to give you more information. But I'm happy to send that to you either through email or LinkedIn. And Jugnu, I
Suresh?
see. Did I answer that?
Yeah.
Yes, you've
it is.
already answered the question. You can go ahead
Yeah.
for other ones.
Looks like Suresh still has his hand up.
Oh, Suresh, yes.
Hi, Madhu, can you hear me?
I can
Yeah.
hear you, yes, Suresh.
Yeah, Madhu, when you said that you did the organoids for SAR and multiple drug screenings, did you also have co-culture? I know the momentum part of it, I got it. But did you have co-culture of other types of organoids or spheroids, like, for instance, a lymphocyte co-culture or something like that?
It's a great question, Suresh. So for the confirmatory screen where we ran it in 1536, we only had two cell types. We had the fibroblasts and we had the epithelial cells. When we did the 96 well culture, that's when we had the adipose cells, we had the resident lymphocytes, and we did the cytokine panel.
So thank you, Madhu. Just to follow up on that one. We are trying to do a co-culture of lymphocytes with the organoid, patient-derived tumoroid, and we have a challenge of ratio of cells needed, like, for instance, T
Yes,
helpers,
exactly.
suppressors, and so on. So I just wanted some insight. Maybe on a one-on-one we can catch up, Madhu.
Yeah, because no, it's a good question to bring up, because Suresh, we've also seen multiple questions come up with activating the resident lymphocytes, matching them to HLA subtypes. So there are multiple different things that, I think we actually just got lucky when we did the screen that many years ago, because now I think there are a lot more things we have to take into consideration, but maybe we can chat offline about that.
Thank you. Thank you, Madhu.
I think, Suresh, that also changes from tissue type and the question you're asking, because we've done co-cultures for immuno-oncology drugs, where we have cancer and PBMCs, and depending on which one we're using, you have to pre-stimulate cells, and again, you have to do a full time course of multiple days to know when you are activating the target cells and seeing the biology you wish to recapitulate. So there isn't a single answer. It's model by model, spheroid by spheroid, target by target, mechanism of action of drug.
Good point, Jagno. Thank you.
I would agree. I see
Hamra.
Hamra's.
Okay. Good afternoon, everyone. Absolutely, a very good presentation. My question is, which genetic makeup from which side? Because we are Africans from Nigeria. Does this study is this study at the end of the study, the drug that is actually found, will it be relevant to Nigerians, or will I say Africans? So it is just limited to the genetic material of that region that was used to model the 3D for the drug testing. And I'm sure this study was done using bioinformatic tools. If you can please mention one or two softwares that were used for these studies. Thank you.
Yeah, so regarding the software, like I mentioned for Kiran Kumar's question, I can definitely send that resource on because it is open source. In terms of the demographic question, that's a fantastic question, Hamra. And actually at Inspiro, this diversity panel in preclinical models is something we are very focused on. I cannot share the information about the 20 patients and the cell lines they use because of the consent forms that they signed, but I can tell you without specifics that we did use a demographic diversity to the extent that those patients came to that particular oncologist's office. So we did have 3 demographics represented.
think Dr. Hafiz has a question. Dr. Hafiz.
Yeah, hi everybody. Madhu, that was an excellent talk. It's nice to know your journey. I have two specific questions. Like say, one is more generic and one is more specific on regulatory
Sure.
interactions. With your experience working with these 3D models, can you share your view on one, like say, the current industry challenges using these alternative approaches? And what are the recent trends in overcoming them? This is more generic kind of a thing, but then I just want to know your viewpoint based upon your experience so far. And the second one is more specific about regulatory viewpoints. And how do these regulatory people look at these alternative approaches, which are, let's say, 3D and both, let's say, microphysiological approaches? And what are the, if you would have to say, let's say, based upon your experience, what are the three important things that they would like to see in these kind of models going forward, right?
Yeah, it's a great question, Hafiz. So I think that I'll answer your second question first because it kind of feeds into the first question regarding, you know, how much people are using this, where they're using this. I think a lot of it is driven by the regulators, and so far, so we actively engage with organizations like nonprofit organizations like the IQ Consortium and the Critical Path Institute, because what they are is they bring the regulators to the table, they bring us vendor method developers as well as industry to the table to be able to share in a pre-competitive space the data that they're generating with these 3D models, not only what works, but what doesn't work. So for example, I was at this cardiac workshop for 3D models at the FDA 2 days ago, and they very clearly said, you know, the reason we go with what we go with now is because we have not seen people submit enough data from the 3D model. So I think the regulators have realized that they need to be engaged. They're making an effort. But I think there's still a little bit of reticence or reluctance maybe on the industry side to show data that doesn't work. So I think it is a work in progress, but I can give you an example. Right now we have a complex in vitro model group, which is truly working to qualify a liver toxicology 3D model for over the next 2 years. And then once that happens, I think that people will adopt it much more wholeheartedly because they know that the data generated by that model will be accepted in an IND.
Thank you.
Did I answer your question?
Yes, absolutely. And would you like to share some of your views on what are the current challenges in these alternative approaches that have been going on, and what are the recent trends in this particular field?
So I can say this is just my sort of humble opinion. I think the challenges come from it's almost cyclical, you know, because the FDA wants to approve things based on the data, but then people always feel they have to do 3 or 4 or 5 other experiments in addition to the 3D models for it to, you know, to submit a package. And I think it's being very cost prohibitive because we're still using all those models to give us the same answer. I think when we get to a point where there's more widespread adoption, people will, you know, we'll stop doing like 5 in vivo pharmacology studies instead of 1 representative one, to be able to, you know, talk about your sort of the efficacy of your drug. But we're not there yet.
Yeah, absolutely. I think, like, you know, I think that resonates my viewpoint on this also as well, because I have had some kind of interactions with them, but then, you know, primarily the main challenges what they have is, you know, the biological variability that the model address. And the other thing would be, like, say, the reliability and reproducibility of these particular models. You know, that is a very big bottleneck. No one wants to put their time and money because it is very resource intensive, actually, these kind of exercises, right? And again, like as you said, regulatory people want to see more data on this. They are very encouraging about it, and they want to see, like, say, what the worst could happen, right? Like, say, they would reject it, right? But then it would, at least, like, say, people will have a wealth of information that can be shared. And I think, like, say, that is also one of the key things what is missing in these particular models is a lot of people are working on this actively, but then they are not sharing the information across, right? And I think recently consortiums are coming up to put up this data, let's say, in a common place so that people can use it. And it is getting there because recently there was one drug, probably, like, say, it went from directly, from these working on these models to clinics, actually, surpassing animal studies. And that is very encouraging. And I think, like, say, it has got a lot of potential. And thank you again for your presentation and, like, say, sharing your views.
Thank you. My pleasure.
We have Alka.
Yeah, hi, Dr. Madhu. Thanks a lot for this very insightful presentation. So I had a question, like, as you mentioned that different cell types were used in the study, correct? Like epithelial cells and fibroblasts. So my question has two parts. So I wanted to know first, with your experience, like, did the ratio of the cells matter in the organoid formation? That is number one. And the other thing I want to know is, was there a specific cell type that was necessary, like, for these organoids to, have the functionality? Like, as you mentioned, it was epithelial and fibroblasts, but I have also like gone through papers and I've done experiments myself that endothelial cells are also something that is people, they do use that. So I would like to know that, did the ratio of the cells that you used mattered, and was a specific cell type which was really necessary for, bringing these organoids together? Yeah.
Those are great questions, Alka, and I will apologize to everyone ahead of time. I have some people cutting down trees behind me, so you can hear that in the background. My apologies. But Alka, great question. So yes, number one, address your first question. The ratio of the mesothelial cells to the fibroblasts and the fibroblasts to the epithelial cells was extremely important. So it was a 1:10 ratio for the mesothelial cells to the fibroblasts, and then a 1 to 250 ratio, I think, no, a 1 to 25 ratio between the fibroblasts and the epithelial cells. And that was based on multiple surgical resections of omental tissue from where from the surgeon who had looked at these over a period of time. So we sort of retrospectively calculated the number of cells that we would potentially need for the assay. So number one, absolutely. Number two, there would have been no metastasis assay without the mesothelial cells. 100%, they were absolutely essential to be able to reconstruct the omentum. When we added the immune cells, when we added the adipose tissue, yes, it certainly augmented it because, the energetics from the fat tissue does accelerate tumor growth. But without the mesothelial cells, it just wouldn't have been there.
Oh, great. Thanks a lot for that.
You're welcome.
Then we have Etinosa.
Yes, good evening, Doc. Thank you so much for your presentation. You have really stimulated me this evening. As a follow-up to the question asked by my Nigerian counterpart about the demographic choice, I know you don't have a liberty to tell us, but did it cut across, especially now that we're looking at personalized treatment? Did it cut across Africa, or did it cut across? That's just my question. I didn't quite get it at the beginning.
Yes, absolutely. And in fact, on a separate note, I can connect with you because I'm on the scientific advisory board for Fight Cancer Global, and we have a project in Nigeria for breast cancer screening, developing biobanks for all of that. So this is something we are, going after with a very focused sort of directive. In the ovarian cancer, what I can tell you was, yes, we had African origin, patients of African origin as part of the patient population. The target, however, We did some transcriptomics on the patient samples. The particular target that the drug was developed against, that was not modified, surprisingly, from demographic to demographic. So the beauty of it would be able to say that at least for this particular subtype of cancer with this target, you should see no change in efficacy. Now, the toxicology and the way a patient metabolizes the drug, that's completely different. But in terms of engagement with the target and efficacy at the tissue of that it's targeting, we did see a difference. So,
Thank you very much. I would like to connect with you so that I can work in
yeah,
that area.
absolutely. Yeah.
Any more questions from the audience?
Hi, Dr. Madhu. Sorry. Hi, Dr. Madhu. Just one last follow-up question that I had, a small one, that you mentioned that ratios with respect to mesothelial fibroblasts and fibroblasts and epithelial. So I just wanted to know, so all these were used together, correct? Like
correct. Yes. Well, we use the mesothelial cells and the fibroblast initially to create the omentum because you have to let them sit for a while to sweep the stromal derived factor, the collagen, this tissue type collagen, let them sit for a while, develop the omental layer, and then we added the epithelial cells.
Okay. That makes sense. I just wanted to, okay, great. Thank you.
Okay, if nobody has questions, we can go to the closing remarks. I'll quickly introduce Dr. C. Damodar Reddy to do this. He is the CEO of Sugen Life Sciences, which is a contract research organization with key focus on preclinical safety, efficacy, toxicology, and clinical research services that they provide to biopharma industries and research organizations. His company provides comprehensive drug testing services involving laboratory-based studies, vertebrate animal models, and clinical studies under regulatory compliance. Dr. Damodar also guides on regulatory compliance aspects in performing in vitro and in vivo studies in small animals and MRI studies in humans, as well as regulatory compliance-related clarifications. He is an advisor and mentor to the executive members of the FABA US chapter. Welcome, sir, and over to you to do the closing remarks. Thank you.
Hi, Deepika, and thank you, everybody. Thank you, Dr. Edenna, and also Srinivas for inviting me. And I was part of this FABA since so long. I congratulate Srinivas and Dr. Edenna for bringing an outstanding speaker, Dr. Madhu Nag. It's a absolutely impressive talk. And my background is biochemist, a cancer biologist, preclinical toxicology company based in Tirupati. I am from Tirupati, and I have been here, and I live in Philadelphia. So I absolutely enjoyed the talk. I hope all the participants enjoyed the talk. The reason is you are at the right spot in terms of the location, excellent expertise, and you have grown from 2D cultures to 3D cultures to INDs, to successful INDs. And also you have a good team with diverse expertise, which starts from bioinformatics, clinical, translational. And the other thing is, in terms of investigative toxicology, which is important, and also you collaborate with NIH. You're part of many of the, you're also occupying some of the positions and connections with FDA, NIH, and also other nonprofit organizations. That means I could not best yet imagine an individual who has a lot of expertise, exposure, 17 years of experience or 20 years, and you're trying to do on your own with a team of people your drug discovery program. Number one, you also try to help globally, internationally, different organizations in a nonprofit way. The other thing is you also collaborate with people who are willing to write SBI or small kind of grants. it's some sort of a consultancy, I think. At the same time, I'm assuming that the assays that you're providing may be available in the as a service mode also. So overall, the activities that your company and the support system that you have, it's excellent. You're doing very good job. And I was impressed by the way you have presented everything. And because we had some interest as a CRO, we are confined to certain tasks only, but we had some interest in this 3D in vivo models. I met Dr. Suresh also about 6 months back, and we have interest. And also this area compared to either diabetes, chronic diseases, and other things, oncology, immuno-oncology, 3D models, you're at the right space. At the same time, as Dr. Hafiz has indicated, impressing the regulators can be a challenging thing. You know what I'm talking about. Most of the audience also know about how difficult it can be because the FDA or the regulators believe in this. But the good thing about FDA people is that they're willing to be open, but they wanted to see a lot more data. So that once you and several others show a lot more data, that will become the basis for them to look at more cases like you in future, that as Hafiz has indicated, without in vivo work, they can upload the drugs to go into the clinic. So with this, I don't want to take more time. I am impressed that there are a lot of questions. I hope your connections with Srinivas, Dr. Edanna, and your association with FABA will be immensely useful. And I wish you all well. And once again, thank you, Deepika and Jagdish, Professor Edanna, Srinivas, for your good efforts. And I'm happy that I'm part of this and continue to be associated with this organization. And again, congratulations for the excellent talk. The first talk is an awesome thing, Srinivas and Professor Edanna. Thank you, Madhu.
Thank you.
All well. We'll be in
Thank you
touch.
very much.
Thank
Thank you very much.
Thank you, Dr. Madhu.
A big applause to Dr. Madhu. Excellent talk.
Thank you all.
Take care, everyone.
Thank you all.
Have
See
a
you in the
good
next
day.
Thank you all.
chapter.
Bye.
There is one
Thanks,
more, I
Madhu.
think, announcement. There is another announcement. Yeah,
Mhm.
go ahead, sir.
So we have the next webinar on June 8th. It's on CAR T cell therapy by Dr. Mohan C. Vemuri. We will share the flyer with you shortly.
Yeah.
Thank you.
Thank you.
Good.
Yeah.
Thank you,
Bye-bye.
everyone.
Have
Thank
a
you.
good rest of the day.
Have a good weekend.
Thank you, sir.
Thank you.
Thank you
Thank you,
all for
everybody.
coming.
Thank you.
Thank you.
Bye-bye.